Good morning to all. So, we are at the types of chromosomes and structure of chromosome are completed in our previous class. The chromosomes are mainly, so they contain the bundles of DNA molecules. So, today we are going to discuss about the next topic, that was about the mitotic division. So, after completion of types of chromosomes and structure of chromosomes, so next we are having the mitotic division. So, these mitotic division, that was mainly about the cell division. So, cell division is compulsory for the development of the cell, the growth of the cell, and also for the genetic information, that is hereditary information, was got transcripted so mainly from one generation to the another generation. So, equally the chromosomes are got distributed equally, and so without any changes. So, those are mainly takes place with the help of mitotic division. So, it was mainly takes place inside the autosomes. The mitotic division was mainly occurred inside the autosomes only. So, inside autosomes, those autosomes are 22 pairs of autosomes are there inside the human being. So, on those autosomes only the mitotic division was takes place. So, in this mitotic division, the chromosome number may not be got deducted. The chromosome number was continued as usually. So, such type of the chromosome. So, after completion of mitotic division, the cells or two daughter cells are formed so with equal number of chromosomes and 2n number of chromosomes. So, today is our topic, that is about the mitotic division today. So, it may possess the some stages and G1 phase, S phase and G2 phase. So, after completion M phase, that is the mitotic division phase. So, the interphase it was mainly occurred during before commencement of mitotic division, some changes are occurred inside the cell. So, those are condensation of chromosomes takes place, and the nuclear envelope disintegration was takes place. So, such changes are occurred in interphase. So, those are first occurred stage, that is interphase. After completion of the G1, S and the G2 phases are there. So, G1 is it was also called as a gap phase. It was the first gap phase, and after it was mainly occurred before occurring of DNA synthesis. So, before commencement of DNA synthesis, the G1 phase was started. So, after completion of a gap phase, so it can take some more time, and after completion of a gap phase, that is a G1 phase, then S phase started. So, these S phase mainly takes place during the DNA synthesis. During DNA synthesis, it was takes place. So, G1 phase it was mainly before commencement of DNA synthesis, and S phase during DNA synthesis. So, that was occurred S phase, and the G2 phase after completion of DNA synthesis. So, it is a G2 phase was after completion of DNA synthesis. The another phase it was considered as a G2 phase here. So, these are G2 phase mainly second gap phase. It is also gap phase, but so the G1 phase is more, it can take some more time than the S and the G2 phase. After the another gap phase is the G2 phase. After completion of these stages, then the mitotic division was started as usually. So, this is M phase was nothing but the mitotic division was started. And now, So in mitotic division, prophase, metaphase, anaphase and telophase. So mainly in after up to the completion of mitotic division, 4 stages are there. So in these 4 stages, so first one is a prophase, and that one is a primary phase, and a metaphase, anaphase and the telophase. Totally 4 stages are there, and we will see the diagram of these 4 stages. First of all, so the prophase. So this is the mitotic division mainly. So interphase, the changes, previous changes are occurred for the commencement of mitotic division. So those are takes place inside the interphase. After completion of that, so G1 phase, S phase and the G2 phases are occurred one by one. So those G1 phase was a gap phase. Why? Because it can take more time. So that should be happened before commencement of DNA synthesis. And the S phase was takes place mainly inside during the DNA synthesis. And the G2 phase after completion of a DNA synthesis. Then after the M phase, the mitotic division phase was started. That should be contain totally 4 stages. So those 4 stages are prophase, metaphase, and that is a pre-metaphase means so the changes are occurred for the commencement of metaphase. So that is a pre-metaphase, metaphase, anaphase, and so telophase. Actually 4 stages are mainly 4 stages are there. So in these first one is prophase. So prophase, in these what happens is so the nuclear material, mainly the chromosomes are presented inside the nucleus. So inside the changes are occurred inside the nucleus, that is actually the chromosomes are not clearly visible. So but for the commencement of a prophase stage, the chromosomes are going to become condensed. The condensation of a chromosome should be takes place, that is the thickening of the chromosomes. The chromosomes are become thickened. So that phenomena was considered as a condensation of chromosomes, and so the centrioles are formed. Centrioles are again divided to form the microtubules. So before commencement of formation of the microtubules, the centrioles are formed nearby the nucleus, and here the nucleolus is also presented here. So here is the nucleolus, and so the nuclear envelope disintegrated, going to be disintegrated means degeneration of nuclear membrane was takes place. So in the prophase, the degeneration of disintegration of a nuclear membrane, that was going to be disappeared, and so the condensation of chromosomes are occurred, and the chromosomes are going to attached with the round shaped bead like structure that is called as a centromere. So centrioles are formed nearby the nuclear envelope. So those are a precursor for the formation of microtubules for the next stage, and these are the changes or takes place inside the prophase. And now pre-metaphase. So the changes are occurred before commencement of metaphase, that is second phase in mitotic division. So in these what happens is the chromosome duplication was occurred. The chromosome duplication, the chromosomes are going to become duplicated, the number was increased. So that's why the sister chromatids, now those are considered as sister chromatids. The edges of the chromosomes are considered as chromatids, those are the sister chromatids. Why? Because the chromosome number was duplicated here, and the centrioles are going to be moved to the opposite poles. In the middle of the centrioles, the microtubules are so slowly, gradually the microtubules are going to be developed. So these are the stages or takes place inside the pre-metaphase, and after completion of the metaphase was directly started. So in the metaphase what happens is the chromosomes are moved to the middle of the microtubules, so middle of the cell. So at the opposite poles what happens is the centrioles are there. In the middle of the centrioles, the microtubules are very well developed. So in the middle of the main middle part of the microtubules, the chromosomes are reached. So that plate was considered as a mitotic plate. So in these mitotic plate, so the chromosomes are arranged mainly at the middle region of the microtubules or the mitotic plate. So these changes are occurred inside the metaphase. Meta means so it is a middle stage. So that's why the chromosomes are reached the middle part of the microtubules or the mitotic plate. So these changes are occurred inside the metaphase. And now anaphase, see this. So see the variation between the metaphase and the anaphase. So centrioles are as usual, but the microtubules are pulled the chromosomes to their opposite poles. The chromosomes are moved to their opposite pole direction. See here, the chromosomes are so in sister chromatid stage, but those are divided and moved to their opposite poles. So in the anaphase, anaphase means the meaning of the anaphase means movement. So that is nothing but the movement of the chromosomes should be takes place. The chromosomes are moved to their opposite poles in here. The chromosomes are gradually moved to their opposite poles with the help of these microtubules, So see here. So these are the changes are occurred inside the anaphase and telophase, that is the end phase. Prophase is the starting stage, telophase is the end stage. In the prophase stage, the disintegration of nuclear envelope, the degeneration of nuclear envelope was takes place here. At the end of the telophase, again the nuclear envelope reformed. The nuclear envelope reformation takes place at the end of the telophase. So what are the cell organelles are going to be disappeared? Those are again reformed in the stage of telophase. So actually, sir, nuclear envelope was going to be disintegrated and those nuclear envelope was reformed in the at the end of the end stage of telophase. So and here it was the chromosomes are mainly moved to their opposite pole direction and in the middle of two cells, the cell was moved and converted to as a furrow was formed in the middle of the cell. So that was a precursor for the deviation of the single cell, individual cell into the two equal parts. So a furrow was nothing but a cleavage furrow. So that was a constriction was formed in the middle of the single cell, individual cell, and the cell was going to be elongated and a furrow was formed in the middle of the so quite a middle part of the cell. Then it was a precursor for it was going to deviate the single cell, individual cell into the two equal parts. So then it was going to divide these individual single cell into the, see here actually, so it was be like this. Two individual daughter cells are formed at the end of at the end of mitotic division, and in these cells the chromosomes are in 2n condition. So there is no changes are occurred inside the chromosome number. So these mitotic division takes place mainly inside the so that was mainly occurred mainly in in autosomes. So autosomes are that was body parts. So the reform, any damages are occurred on the surface of the skin region or the inner region of the body. So those damages are going to be repaired with the help of these cell divisions. The cell divisions are mainly helpful for the so those are reformed. So damaged parts, damaged cells are reformed through the cell division, that is only the mitotic division. With the help of mitotic division, the damaged part, the damaged organs are reformed with the help of these mitotic division. So in this mitotic division, a single individual cell was finally divided into two equal parts. Two individual daughter cells are formed at the end of mitotic division. So the importance of mitotic division was it plays a major role in cell division. So it was mainly helpful to reform the development of the cell, the growth of the cell, and that was mainly there any damaged part, any so repairings are mainly occurred inside the body or outer side of the skin. Also any damages are occurred, any wounds are occurred, so those are again repaired with the help of these cell divisions. Again newly formed skin region was again reformed at that area. The damaged part was again reformed with the help of these cell division. That is nothing but mitotic division. So after completion of mitotic division, you are having in your syllabus, that is another topic that was about the meiotic division. The meiotic division was reduction division. The chromosome number was going to be reducted. So that was here 2n conditioned chromosome daughter cells are formed inside at the end of mitotic division, but at the end of my meiotic division, the chromosomes are n-numbered chromosomes, daughter cells are formed at the end of meiotic division. The meiotic division only takes place inside the male and female gametes, that is a sperm cell and ova only, but not in autosomes. Inside the autosomes, mainly inside the human beings, 22 pairs of autosomes are there. So those are mainly helpful for the body organs or any tissues or any organs are going to become damaged, and so those damaged parts are and the development of those organs. So at that time only these cell division was takes place. These cell division was very helpful, essential for that, but not the meiotic division. The meiotic division only occurred mainly inside the male and female gametes only. So that should be helpful for the formation of n-numbered condition of daughter cells are formed at there. Here 2n number conditions of chromosomes are formed here. So n-numbered chromosomes and 2n number chromosomes that is the main variation. The n-numbered chromosomes are furtherly helpful for the formation of the gamete. So the 2n conditioned cells are helpful only inside the body parts. The remaining, the gametes, are helpful inside the at the end of. So these are for the zygote formation. So this division was very helpful. So it is about the mitotic division. So today's topic is over. We'll meet tomorrow with new topic, that is the meiotic division. Today's topic is over. We'll meet tomorrow with the mitotic division. So mitotic division was mainly in body cells, and the meiotic division was only inside the gametes. And so in prophase, subphase are there inside the meiotic division. That is a leptotin, zygotin, pocketin, diplotin, diakinesis. Those substages are not presented inside the prophase of the mitotic division. Those are takes place only inside the meiotic division subphase or prophase subphases. But those are not visible inside the mitotic division, and the mitotic division was occurred only inside a single phase, but meiotic division was takes place that was up to the 2 stages. So this is our today's topic. Today's topic is over. We'll meet tomorrow. So we will meet tomorrow with a new topic. I am going to stop here. Thank you.